And we're back. Mr. Kleiman here with more grade 12 biology. We've just finished studying DNA replication, and now we're on to the central dogma of molecular biology. And that is going from DNA to RNA to proteins. And in today's video, we're going to focus on transcription, going from DNA to RNA. How is it done? Let's dive right into it. There is just one incredible enzyme that does most of the work of transcription, and it's this beauty right here, and it's called RNA polymerase II. And what you can see it doing in this picture is unzipping DNA and copying one strand of it into RNA. We call that process transcription, and that's the first part of the central dogma of molecular biology. So when I say the central dogma of molecular biology, again, that's DNA to RNA. That's what we're focusing on this video. And I'll be making a second video that shows you how we go from RNA to protein. We call that process translation. Some people also include this step in the central dogma, and we've already covered that in a previous video. And that, of course, is DNA replication. So here's the big idea. We're going to take our DNA template, which holds all the instructions to make every protein in your body. We're going to convert them into RNA molecules, and we're going to read those RNA molecules 3 letters at a time to spell out which amino acid to add in which order in order to create a protein. So this begs the question, why the intermediate? Why don't we go directly from DNA to protein? We need to make proteins at the right place at the right time. And so that means that we don't want to have every single protein being made everywhere all at once. We want to have the control to only create the proteins we need where we want them and when we want them. So think of a prokaryotic cell which has just DNA and some ribosomes around it. If those ribosomes bump into DNA and immediately start making protein, then any gene on that entire genome is going to start getting made at any time. Instead, what we want to do is to only copy the instructions to make the gene we want. And that's why we make the RNA transcripts. Ribosomes do absolutely nothing when they touch DNA. Instead, they only get kind of turned on and start to work when they bump into a molecule of RNA. That intermediate gives us control. When you think about a eukaryote, all of their DNA is locked up inside of this nucleus. And if you recall, one of the biggest challenges that all cells have faced is that they're always under attack by other creatures. In particular, our prokaryotic ancestors were being attacked by viruses. And a virus works by injecting its own DNA into your cell and trying to take it over. And so one of the defenses that eukaryotic cells created is to bundle up our DNA into a bubble. And now any DNA that you would find in this region here, the cytoplasm, doesn't belong, and so you can chop it up. Or you can keep your DNA protected in the nucleus and send out only RNA. RNA belongs in this space, and any foreign DNA is going to get chopped up. Now, of course, I'm sure you know some of the rest of that story. Viruses have adapted, and many of them have learned to inject RNA rather than DNA over the millennia. But this is most likely one of the selection pressures that led to the creation of the nucleus. And after that nucleus was formed, we have yet another benefit of making RNA and having this nucleus. And that is that now when we make the RNA copy, which will be inside of the nucleus, there's a time delay. There's going to be a lag between when you make RNA and when it actually reaches the ribosomes. And what we're able to do inside the nucleus is actually modify those RNA molecules in meaningful ways so that from now a single gene, one set of instructions in the DNA, you can actually make a whole variety of different RNA molecules, which will ultimately result in a whole variety of other kinds of proteins. And so now a single gene isn't really just coding for one protein, it actually codes for a whole family of protein. So here's the overall picture of that story. We're going to go from DNA to RNA. We're going to be able to modify that RNA inside of the nucleus. Then we're going to send it out of the nucleus through this nuclear pore into the cytoplasm as a form of messenger RNA. And when that bumps into a ribosome, the ribosome is going to translate that into protein. Messenger RNA is an exact complementary copy of a specific gene, and that's the version of the nucleic acid that can leave the nucleus and reach the ribosomes. And so remember, if we're going to be making a specific protein at a specific time, step one, as it was for DNA replication, is that first we need to uncoil the region of DNA where that specific gene is located. So once it's uncoiled and we have access to it, there are 4 phases of transcription. And so there's the initiation phase, elongation, termination, and if you're a eukaryotic cell, you can even do some further processing if you've got a nucleus. And so there's that RNA polymerase once again. And so RNA polymerase is going to bind to the outside of that DNA molecule with the help of some proteins called initiation factors. And then it's going to have its own built-in helicase activity where it's going to unzip the DNA just inside of the enzyme itself, add in RNA nucleotides and complementary base pair with the template strand, and create a perfect separate transcript of that gene. So at this point, you should pause the video, go to the description at the bottom, and have a look at how RNA polymerase binds to the correct region of DNA. It's really quite beautiful and fascinating. Alright, so hopefully what you saw in that video is that RNA polymerase is going to get some help finding the right location of where genes are. And so on the actual DNA itself, you'll have a region of the DNA that's usually rich in thymine and adenine, that's called a promoter sequence. And the promoter sequence, sometimes called the TATA box, is a location where an initiation factor or a transcription factor will bind. And this thing will sit on the outside of a fully coiled up DNA molecule. When RNA polymerase II bumps into that initiation factor, a whole bunch of other transcription factors start to assemble. And when they're assembled just right, now we're ready to begin transcription. And so you can see that that helicase activity is starting when all these factors are assembled correctly. The physical distance between the promoter and the gene is actually going to tell it where to start. Some of these, like you saw in the video, require even an additional step beyond the promoter. So here's the promoter where you'll have that initial binding of RNA polymerase. Some of them even farther downstream will have what's called an enhancer, a protein sitting there and waiting, and it must be the final component to attach to this complex of RNA polymerase with all of those transcription factors and send it off running. And so those promoter regions are going to be upstream of the gene, so just before the gene. They're going to determine exactly where RNA polymerase binds. They're going to determine exactly where transcription therefore begins, and they're usually rich in thymine and adenine, so we call them the TATA box. Here's an example of some promoter regions in E. coli. And so this +1 location is where transcription actually starts. This is the beginning of a gene. And if you look just downstream of it, you see these 2 regions here rich in thymine and adenine, and these are the general sequences where those transcription factors are going to bind. These parts are variable. This is the binding sites for those transcription factors. So one of our strands of the unzipped DNA is going to be a template, and the other one is not. And so which one is going to be which? Well, it's going to depend on the 3D orientation of RNA polymerase. The first thing that's going to happen is that helicase will unzip that DNA molecule. And now it's going to start to add RNA nucleotides through complementary base pairing. And so once again, let's remind ourselves that the main difference between an RNA nucleotide and a DNA nucleotide is that this is a different sugar. This S here represents a ribose sugar, and in a DNA nucleotide, this would be a deoxyribose ribonucleic acid. So this is deoxyribose sugar. Let's also notice the direction that this is moving in. If this is the 3' end of the template strand, well, this must be the 5' end of the complementary RNA strand being created. once again, directionality is everything. Once this thing gets oriented correctly, once RNA polymerase is oriented correctly on your gene, it's always going to move in the 5' to 3' direction. In other words, the free 3' end of this sugar is where the binding of the next nucleotide is going to be. And so we call the other strand the coding strand, and that's because the coding strand is identical to the code that you see in the mRNA. The template will be copied, and its complement is the instructions to make the protein. So here you can see all of it in a single diagram. Here's RNA polymerase working its way down that DNA strand, adding RNA nucleotides as it goes, and you can see the DNA winding itself back right behind the enzyme. No single-stranded binding proteins needed. Here's another diagram of the same thing. You can see the unzipping and the rezipping of DNA, and in the middle we're adding RNA nucleotides. Okay, out pops this molecule of RNA, and the reason why it's able to come off so easily is because that difference in sugar and the use of uracil rather than thymine makes the binding of RNA much weaker. Okay, so mRNA is being transcribed in the 5' to 3' direction. During this phase we call elongation. This is actually making the mRNA transcript. DNA unwinds only in the regions where transcription is occurring, so within the RNA polymerase molecule, and after transcription the DNA coils itself back up. What's really cool is that you can have lots of RNAs working on a single gene at the same time. So once you've unfolded the correct region of DNA and you've attached your transcription factors, multiple RNA polymerases can come and start binding and going. So here you can see it's sort of like a highway. This is like traffic. You've got 1, 2, 3, 4, 5, 6 of them in a row, headed in this direction, going the 5' to 3' direction, and the farther along they are, you can see the longer the RNA copy is that they've made. And as soon as there's room for a new one, they'll continue to bind and bind and bind until this process is done, until this gene is no longer needed and we turn it off. So here's an actual electron microscope image of actual bacterial DNA being copied. And you can see the beginning of that gene, where you've got very small RNAs coming off. And here you've got the end, where you've got these really large bits of mRNA coming out. And so all along this gene, you've got RNA polymerases, and you can see multiple genes back to back to back on this DNA molecule, all being synthesized at the same time. So this region of the DNA is active. Now we're going to get into the final stage. How does it stop? How does it know where the end of the sequence is once this thing starts flying through? And we call that termination. So a terminator sequence in the coding strand tells RNA polymerase exactly when to stop transcribing that mRNA. And it's going to cause RNA polymerase to be released. And we can reuse it once again. It can make another copy over and over again. And that mRNA is now released into the nucleus. And so all it is a sequence on the DNA itself. We call that a terminator sequence. And the reason why that causes RNA polymerase to pop off is actually because as soon as that sequence forms, it folds. Okay, so that region right there is this region right over here. And you can see that very quickly it can fold on itself, and we can complementary base pair the RNA to itself. And it creates this little loop that pops off RNA polymerase. Now, if you're a eukaryotic cell, you now have a copy of that mRNA inside of the nucleus. There are no ribosomes there. We're not making protein yet. We've still got a bit of time. Before we export it, we have the opportunity in eukaryotic cells to change that gene into something similar, but a little bit different. And so in eukaryotic cells, the RNA transcript is called pre-mRNA when it's in the nucleus, because it still can be modified before it leaves the nucleus. And why would you ever want to do that? What we're going to do is remove introns, which are these non-coding regions in strategic ways so that we can make a whole family of proteins that are similar but slightly different from each other. We can also add things to that mRNA molecule that are going to determine how it behaves and how long it lasts in the cytoplasm. So that original gene sequence has some regions of that gene that are meaningful and code for functional protein, and some regions that code for nothing, that are nonsense, that need to be cut out of that RNA in order for it to work. We call the coding regions exons, we call the non-coding regions introns, and a huge chunk of your DNA actually codes for nothing. Most of your DNA, in fact, is introns, and only a very small percentage of the nucleotides you have in your cells are actually exons or coding regions that determine the order in which to assemble amino acids. And so let's have a look at how this works. Okay, if that's your DNA with an interspersed sequence of exons followed by introns and more exons and introns, we're going to start transcription here. We're going to end it at the end of the last exon, and then we're going to splice out the introns accordingly. Okay. Once you put together all the exons, this final sequence right here is your final mRNA transcript that's ready to go to the cytoplasm. But look what you can do. Okay. You can make these little snRNAs, and if you remember that, that's just one of the versions of RNA that has some enzymatic activity made in the nucleolus. Okay. This small nuclear RNA can grab on to specific introns, okay, and cut them out very specifically. And if you manufacture different snRNAs, you're going to cut out these introns in slightly different ways. An snRNA binds here, another one binds there, several others form a complex. This creates what's called a spliceosome. Okay, and by the way, the yellow regions are protein and the blue regions are made out of RNA, and so together they make what's called a spliceosome that's able to cut out this region and stitch together the exons. Here's the same starting pre-mRNA, but you can get two different final mRNAs depending on which snRNAs are present. In other words, depending on how you splice it. If you take out intron 1, 2, and 3 in order, okay, you splice them out, you end up with exons 1, 2, 3, and 4 all linked together. But if you only make the snRNAs to grab here and here, but not here and here, you can cut out this entire region with exon 2. And in the end, you'll get a very similar protein, but without the region where exon 2 was, a shorter, smaller protein in the same family as the protein you'd make from this mRNA transcript. Okay, so this gives you a little bit more control over the final proteins you're going to make from a single gene. And so here you can see it a little more visually. Here you can see 3 different versions of proteins created from the same original DNA sequence. And you can see that they're each missing different regions. This has all of the exons from 1 through 5. This one is missing region 3, which corresponds to this loop of the protein right here. This one is missing number 4, which corresponds to this loop right over here. The final step in processing is to add a cap and a tail. So if this is our mRNA after splicing out the introns, this is just the exons that are left. On that mRNA transcript, we can add a cap made out of modified guanines, and we can add a tail, which is just a long sequence of adenines. Okay, sometimes we call that the poly-A tail, meaning many adenines. The cap is going to be there to allow the mRNA transcript to bind correctly to a ribosome. And the tail is going to determine how long this mRNA lasts in the cytoplasm, because it's going to protect it essentially from restriction endonucleases. These are the molecular scissors that are cutting up foreign DNA in the cytoplasm. And essentially they're going to chop up the tail before they get to the meaningful parts of the gene. And the longer the poly-A tail, the longer the mRNA tends to last. And so there it is, pre-mRNA, and here's our final mRNA with both a cap and a tail, introns removed. And so let's do a super quick comparison of how transcription goes in a prokaryotic cell, like this bacteria, versus a eukaryotic cell, like this one shown on the right. In a prokaryotic cell, all you have is DNA, and surrounding it in the cytoplasm are ribosomes. RNA polymerase will unzip the region of the DNA that's needed to make the correct protein, read that gene and turn it into mRNA, and immediately ribosomes will bind to that mRNA and right away start turning it into protein. In a eukaryotic cell, when that RNA transcript is being made, the mRNA contains introns, and we can do some alternative splicing to make different versions of this molecule. We can also add a cap and a poly A tail. So here's our pre-mRNA, and here's our final mRNA. Now this RNA is free to leave the nucleus, where it's going to interact with ribosomes, which are going to translate it into proteins. So overall, the eukaryotic cell is able to create a bigger variety of proteins from the same number of genes. This is a good place to pause the video and follow the link in the description to watch 3D animation of the entire process of transcription. And so here's where we are. We've just learned about transcription. And I wish I could take credit for this picture, but I found it on the internet. So here you've got our hero of the day, RNA polymerase, that's unzipping, copying, and letting the DNA coil up behind it. And in my next video, we're going to look at this monster, the ribosome, which is going to be able to read that mRNA transcript and assemble the correct polypeptide chain to make a protein.